Hello and welcome back to the Neuron Regeneration Podcast. My name is Jared Carter, and this is going to be one of our abstract and paper review recordings. We are looking at the latest in papers and news in regards to neuron regeneration. More specifically, a lot of what we read in these episodes in particular come from a PubMed search on neural regeneration, neuron regeneration, or nerve regeneration. And we look at the most recently published articles that seem relevant to our topic. Before we dive in today, I do want to make a quick correction from the intro, or episode number 1 of this podcast. I was unfortunately mixing up neuroregeneration.org as well as, mixing that up with the International Symposium for Neural Regeneration. Those are two different things. The International Symposium for Neural Regeneration is the conference that I mentioned, and which I actually attended in 2023, and it's fantastic. They hold it every other year. So if you're interested in details on that or potentially attending in 2025, I hope to be there again. You'd go to www.theisnr.org and then neuroregeneration.org. That is the site of Dr. Richard McCurtry, who's an accomplished physician, surgeon, scientist, and engineer. Lots of great information there on neural regeneration. And so I would definitely strongly suggest you check that out if you are interested in this topic. Okay, with that said, let's get into our paper abstract readings. So first, paper today is from Journal of Nanobiotechnology from Pan et al., or maybe Pan et al. Again, I'm just going to pre-apologize for mutilating not just names, but also scientific terminology in these. But again, you'll get the point. This is going to be a great way for anyone interested in this topic to stay up to date on the most recent publications and news on the topic. So, implantation of biomimetic polydopamine nanocomposite scaffold promotes optic nerve regeneration through modulating inhibitory microenvironment. Okay, and here's the abstract. Optic nerve regeneration remains challenging worldwide due to the limited intrinsic regenerative capacity of retinal ganglion cells, hereafter RGCs, and the inhibitory microenvironment. Oxidative stress induced by excessive reactive oxygen species, or ROS, following optic nerve injury is associated with prolonged neuroinflammation, resulting in a secondary injury of RGCs and the impairment of axon regeneration. Herein, we developed a bionic nanocomposite scaffold with immunoregulatory ability for enhanced optic nerve regeneration. The ice templating method was employed to fabricate biopolymer-based scaffolds with a directional porous structure, mimicking the optic nerve, which effectively guided the oriented growth of neuronal cells. The incorporation of bio-inspired polydopamine nanoparticles further confers excellent ROS scavenging ability, thereby modulating the phenotype transformation of microglia and macrophages from pro-inflammatory M1 to anti-inflammatory M2. In a rat optic nerve crush model, the implantation of the scaffold enhanced survival of RGCs and promoted axonal regeneration. Our study offers novel insights and holds promising potential for the advancement of engineered biomaterials in facilitating optic nerve regeneration. And as you'll hear, it seems a lot of the recent publications have to do with scaffolding or other bioengineered materials. Okay, next we have from the Journals of Otology, Rhinology, and Laryngology, the effect of I'm going to butcher this medication, rosuvastatin, on facial nerve regeneration after facial nerve injury, an experimental animal study by Denser et al. So, abstract objectives. Rosuvastatin is an anti-hyperlipidemic statin group pharmacological agent with antioxidant, neuroprotective, and anti-inflammatory effects. In this study, we aimed to examine the functional, electrophysiological, and histopathological effects of rosuvastatin, or in combination with corticosteroids, on facial nerve regeneration in rats with traumatic peripheral facial paralysis, and hereafter that's PFP. Methods. PFP was induced in 28 female Sprague Dawley rats that we divided into 4 groups. Group 1, control group. Group 2, methylprednisolone group. Group 3, rosuvastatin group. And Group 4, the rosuvastatin and prednisolone group. Electrophysiological, functional, and histopathological examinations were performed before and after the medications. Results. Electrophysiological threshold values of group 3 and 4 were found to be significantly lower than the control group on day 21 after treatment. In histopathological evaluation, axonal degeneration, macrovascularization, and vascular congestion levels were compared between the groups, and a statistically significant difference was observed in group 4 compared to the control group. The recovery time of the eye corneal reflex was found to be significantly higher in the control group than in the groups 3 and 4 when comparing postoperative day 1 to day 7 and postoperative day 1 to day 14. Conclusion. Rosuvastatin, especially when combined with prednisolone, was found to significantly increase the facial nerve electrophysiological, functional, and histopathological recovery in injury-induced traumatic PFP. Next paper from the Journal of Colloid and Interface Science by Zhang et al. Clickable immune microenvironment modulated hydrogels for spinal cord injury repair. Spinal cord injury, or SCI, is a devastating condition without effective therapy currently available. The inflammatory cascade following SCI leads to neuronal apoptosis and glial cell activation. The utilization of local injectable hydrogels with immunotherapy drugs directly into injured nerve tissues represents a promising therapeutic strategy. Herein, injectable hydrogels grafted with clickable, there it is again, methylprednisolone. I'm just going to say prednisone. You guys, you'll know what I'm saying. So herein, injectable hydrogels grafted with clickable prednisone and cellular adhesion peptide were developed using free radical polymerization for promoting nerve regeneration following SCI. Prednisone conjugated hydrogels could modulate the immunoinflammatory microenvironment of SCI and sustain neuron survival. The multi-stiffness hydrogels were fabricated by adjusting concentration ratios to evaluate appropriate mechanical stimuli. In a model of dorsal root ganglion, prednisone grafted hydrogels with mechanical signals similar to those of adult rat spinal cords demonstrated superior efficacy in promoting dorsal root ganglion growth. Prednisone grafted hydrogels could regulate the immune inflammatory microenvironment, promote recovery of both motor function and sensory functions. The positive findings suggested that the interplay between immunomodulation and mechanical signals plays a crucial role in promoting nerve regeneration, indicating significant potential for hydrogels as a therapeutic approach for repairing SCI. Next paper from the Neural Regeneration Research Journal. Olfactory Receptors and Neural Regeneration in the Central Nervous System by Franco et al. Abstract. Olfactory receptors are crucial for detecting odors and play a vital role in our sense of smell, influencing behaviors from food choices to emotional memories. These receptors also contribute to our perception of flavor and have potential applications in medical diagnostics and environmental monitoring. The ability of the olfactory system to regenerate its sensory neurons provides a unique model to study neural regeneration, a phenomenon largely absent in the central nervous system. Insights gained from how olfactory neurons continuously replace themselves and reestablish functional connections can provide strategies to promote similar regenerative processes in the central nervous system, where damage often results in permanent deficits. Understanding the molecular and cellular mechanisms underpinning olfactory neuron regeneration could pave the way for developing therapeutic approaches to treat spinal cord injuries and neurodegenerative diseases like Alzheimer's disease. Olfactory receptors are found in almost any cell of every organ or tissue of the mammalian body. Didn't know that. This ectopic expression provides insights into the chemical structures that can activate olfactory receptors. In addition to odors, olfactory receptors in ectopic expression may respond to endogenous compounds and molecules produced by mucosal colonizing microbiota. The analysis of the function of olfactory receptors and ectopic expression provides valuable information on the signaling pathway engaged upon receptor activation and the receptor's role in proliferation and cell differentiation mechanisms. This review explores the ectopic expression of olfactory receptors and the role they may play in neural regeneration within the central nervous system, with particular attention to compounds that can activate these receptors to initiate regenerative processes. Evidence suggests that olfactory receptors could serve as potential therapeutic targets for enhancing neural repair and recovery following central nervous system injuries. Very interesting. And this does look like it has a full text link. This is probably one I'll dig into myself. And as a reminder, with all of these types of episodes, you can always go to the show notes for quick access to all the papers that I'm reading. And the show notes you can find at neuronregeneration.com / and then the number of the episode. So that would be /5 in this case, not written out, but the actual number. Okay. And our final paper of the episode and this was an interesting, I don't know if it's a journal, I'm guessing, but it's Cureus, but it's Cureus. And the title is Alginate Use in Orthopedics and Peripheral Nerve Repair, a Systematic Review. And this is by Matthew Eisenberg and Joshua Husted. The use of alginate, a derivative of seaweed, has been proposed for multiple orthopedic indications. We aimed to review the current use of alginate in orthopedics and to focus on the future applications of alginate for peripheral nerve repair. A comprehensive literature search was performed to identify biomechanical, laboratory, animal, and human studies where alginate has been utilized for orthopedic or nerve repair indications. A systematic review of orthopedic indications was conducted for safety and efficacy, and a specific focus was placed on alginate for use in peripheral nerve repair and reconstruction. 32 studies were identified. Alginate has a strong history and safety profile for usage in orthopedic surgery. Its primary usage has been for the repair of articular cartilage, although it has been used for disc regeneration of the lumbar spine and for cushioning joints in osteoarthritis. The primary indication in peripheral nerve repair is to create an environment that encourages Schwann cell migration and repair in nerve injuries while blocking fibrotic scar tissue formation by inhibiting the activity of fibroblasts. Alginate hydrogel may serve as a potential conduit for nerve regeneration in nerve injuries with small to medium-sized gaps. Very interesting. It seems that there is a kind of a theme for the episode today of bioengineered gels or scaffolds or a mix of both. So there you have it, folks. Again, neuronregeneration.com /5 if you want to access any of these papers. Deeply appreciate you tuning in, and we will see you in the next one.